So for the final parts of our conference, we're going to use a slightly different format and move on to 2 panel sessions. If the panelists for the first session would like to make their way forward, we're going to invite representatives of the pharmaceutical company to come and take a seat. You'll find your names on the seats. And hosting the panel will be Emily and Professor Volker Straub.
Do you want a mic as well? I'll get you a mic as well. Yeah. Okay.
Great. So everyone found their seat. That is excellent. Actually, this is in alphabetical order, and we didn't consult Professor Montoni. So you will be pleased that it should actually work. And so far we have heard from researchers and healthcare professionals. We just had the patient voice as well when Nick Catlin and Janet Hoskin had their educational session. But now we come to an important stakeholder group, of course, as part of this community as well, the pharmaceutical industry. And it's actually, I think, amazing to have 10 companies here that are all involved in drug development programs for Duchenne muscular dystrophy. So thank you very much for agreeing to this, because you can't take this for granted. The pharmaceutical industry is, for understandable reasons, highly regulated, particularly when it comes to interaction with patient advocacy groups, with prescribers as well. There is a code of conduct that is regulated by the Association of the British Pharmaceutical Industry. And so the colleagues have to apply to these rules. They nevertheless agreed that we can have this session, but one prerequisite was that we inform them about, okay, these are the questions that we have, and they also have to then let us know, okay, these are probably the answers that we will give. If there are any questions from you about actually how the code of conduct or code of practice for the Association of the British Pharmaceutical Industry works, then we also have Will Pender, who is the Senior Policy Manager. He's in the background there. He's very happy to answer any questions about this. The way we organize the session is we have about 14 minutes. We will ask each of the colleagues here a question. You will actually also see the question on the screen. And they have about 2 minutes to answer. We will ask the company, and I would be grateful if you could then actually respond also with your name and your job title. Don't spend too much time on that, and then you come to your... Yeah, I mean, sometimes the job titles in pharmaceutical industry, I can tell you. And then you come directly to the answer, okay? Well, maybe we'll just start, and I'll start with the colleague actually from B, with BioMarin. And the question is that BioMarin has been involved in DMD drug development for quite some time. And so what are the key lessons you have learned, and what do you think are the biggest challenges in developing a drug for Duchenne muscular dystrophy?
Thank you for the
Yep.
Thank you, and thank you for the question. My name is David Neal. I'm a clinical development team lead at BioMarin, and I'm developing drugs to hopefully treat Duchenne muscular dystrophy. So with regard to the question, I think one of the most important lessons is, of course, the importance of the patient voice. We've heard this in talks this morning. There are great many effects that the patient voice can have through patient advocacy communities, and we can incorporate those into our clinical trial protocols, be it with regard to patient inclusion-exclusion criteria, the choice of endpoints, what do patients really want with regard to transformational medicine. And with that, I think that's one of the greatest things. We sought advice from the World Duchenne Organization Community Advisory Board for our ongoing clinical trial protocol, and incorporated many of those lessons. And pleased to say that it had a huge deal of impact in how we thought about our clinical trial design. In terms of challenges, I think we've heard some of those this morning. We've heard about the variability in disease progression between patients, and that can make detecting treatment effects really quite difficult. You couple that with the less than optimal clinical trial endpoints that we have currently had available. I think this has made drug development really quite difficult. However, I'm really excited with some of the newer endpoints we see, the digital health technologies, improvements in imaging, and I think that gives us real optimism for the future. So thank you.
Yeah, thank you very much. I would agree that from a clinician's point of view, we definitely have seen a shift more to patient-focused outcomes and involvement as well, which is great. Over to you.
Question 2 to Dine. Young people have now been treated for a number of years with various dystrophin replacement therapies. Can you comment on the evidence of dystrophin protein expression in human muscle and its potential clinical effect?
Sure. Hi, I'm Ash Dugar. I'm the Chief Medical Affairs Officer at Dyne Therapeutics. I know I have 2 minutes, but I'm going to use a little bit of that time for one quick story. I met Emily and Alex, 9 years ago, first 2 advocacy leaders, first 2 moms I met. And when they left, I was like, holy hell, what a force. And clearly, you continue to be a force, and this meeting is amazing, so thank you. It's a great question. You've heard from Professors Artz-Marus, Professor Straub, Montoni. That was not in alphabetical order in terms of some of the exon skipping technology, dystrophin production, and sort of the therapies that are coming around for having even better exon skipping and dystrophin restoration. So maybe there's 3 things I would answer with respect to this question. One is dystrophin continues to be sort of the underlying issue with this disorder. And so there are a couple of methods that companies use to measure dystrophin that they've been using for a long time. Western blot, where you look at the total protein, and also imaging, such as the image that Professor Straub showed, when you look at how much dystrophin is at the muscle membrane and you quantify dystrophin-positive fibers. So that's really important. What's been lacking in the field is understanding the change in dystrophin over time as it correlates to the change in function over time. And so those longer-term studies, real-world studies, are really needed to be able to understand what's happening over time. So what we do know from the literature is the data's out there with different levels of dystrophin. There are differences in terms of disease progression. And so it's actually established in that sense of if you can have 0 versus 5 or 0.5 versus 5, you're going to see different time to loss of ambulation, for example, or different slowing of respiratory decline. And so the data exists that being able to increase dystrophin over time will lead to clinical benefit. It's just that you are still slowing progression. And so hopefully next generation therapies can actually show a slowing or even improvement.
Thank you very much. We'll come to E, like edgewise. And so the question is, edgewise is involved in drug development programs for Becker muscular dystrophy, which is an allelic disorder to Duchenne. We haven't really talked much about it. But what is the experience that you can bring from Becker muscular dystrophy trials to the Duchenne field?
So first of all, I'm Abby Bronson. I have one of those pharmaceutical titles that doesn't really make sense. I'm the head of patient engagement and external innovation. So if you can figure out what that is, tell me what it is. So, but to answer the question, so at Edgewise, yes, we've gone into Becker first, and we really look at Becker and Duchenne as a continuum because the underlying mechanism of disease, muscle breakdown, is basically the same. It's just that the rate of progression in the 2 ends of the spectrum is different. In both diseases, muscle damage is caused by contraction-induced injury. I mean, you all remember your kids when they were born, they had no dystrophin, but they were walking around. So it's not dystrophin that creates force in the muscle. It's dystrophin plus contraction that causes the damage. So our approach in Becker and Duchenne is to prevent this contraction-induced damage by targeting muscle and fibers. In fact, there's susceptible muscle fibers, those that break down more quickly, and those are the ones that we're trying to modulate and provide protection. So, there's a couple things I want to talk about in terms of what we've learned from Becker. And the first one's a real advantage for you guys in Duchenne because, we went into Becker first. We learned so much that we can, and we're continuing to learn. We learned, sort of our fundamental thesis, how can we modulate muscle to provide protection during contraction-induced injury? how the drug is absorbed and moves through the body and eliminated. That was really important. We heard earlier about compounds having to get into the cell, into where it needs to work, and so we were able in dystrophic tissue in Becker see how our compound got into the muscle, and that's where it needs to be. And we also learned a lot about safety, and this is, I think, the benefit for you, because not only have we been able to accelerate our timelines into Duchenne because of these learnings, but we've also been able to, if we had gone into Duchenne first, there have been multiple and longer safety visits in the beginning of the trial because we're very concerned safety first. Because of our learnings in Becker, the Duchenne trial that we designed, it's a little, it's more patient-centric, shall I say. So we really tried to reduce the patient burden in our trial designs. And I guess the other thing that I'll just briefly mention is, we talk about dystrophin all the time. And I remember when I was first in this field in 2009, I didn't even know where the ends of dystrophin connected to, right? We learned about 79 exons, RNA, DNA, exon skipping. And so to understand our approach, you have to really understand sort of like the contractile machinery of how force is created. And so it's a really new concept. And by talking a lot in Becker and kind of figuring out how do we really communicate the mechanism of action or the mechanism of disease, and then what we're trying to do, that really helped us a lot as well. So I've actually really enjoyed working in the Becker field.
Abby, you need to come to...
Oh, thank you very much.
Thank you, Abby. Question 4 is to ITF Pharma UK Ital Pharmaco Group. How do you ensure you engage and involve patients in the development of new treatments?
Thanks, Emily. Good afternoon, everyone. So my name's Roger Berry. I'm the medical director at ITF Pharma UK. So we're a newly formed biotech company in the UK. We're part of the wider Ital Pharmaco Group. A couple of my colleagues are here, Ross Selby and Sam Parker. So we're honoured and humbled to be here at this conference, and I echo Ashley's sentiments. Huge congratulations to the organising committee. A really incredible day. So we believe that patient involvement in everything that we do is absolutely paramount. So we strive, to build partnerships with patients and engage them throughout the whole drug development pathway. So right from the very early ideas of a new drug through to clinical trials and then into the post-marketing and approved setting. So some examples of that. So during our early clinical trials, our research and development team worked with the DMD community across Europe to really optimize the trial design for our drug, but also to optimize the endpoints that we used in our trials. And furthermore, they validated a lot of the patient-facing materials. So things as simple as testing that the materials are easy to understand or they're readable from a patient and a carer perspective. We're very much used to, as an industry, using too much technical language. So moving forwards, we're committed to continue that patients are at the heart of everything that we do, and we'll do that in 3 main ways. So 1, continue the great collaborations with the patient advocacy groups and the charities, attending conferences like this so that we can learn from you as a community. Number 2, focus on the measurements in trials that mean most to patients. And thirdly, by organizing and delivering advice-seeking sessions from yourselves to help us when we develop our medicines from the beginning through to the end. So asking you questions and asking for input. So we look forward to continuing to collaborate with you as the community and really helping to improve the lives of the DMD community.
Thank you very much. Our next question is to Pfizer. Please could you explain how you use patient data in the development and approval of new treatments, commenting on what data is most useful for what purposes?
Thanks, Emily. So my name's Ione Willacott. I'm a Senior Medical Affairs Manager. I work within the Rare Disease Specialty Care team at Pfizer UK. So during drug development, clinical trials need to comprehensively assess a treatment's safety and efficacy while generating data that are actually meaningful to patients and their families and complying with data governance laws. So at Pfizer, we involve patients really early on in our trial design, and we also ensure we have strict standards of data privacy, security, and quality. So we're using technology to gather patient data in more meaningful and less burdensome ways in our trials. For example, wearable devices are being used to collect continuous movement data in participants. And these can measure the potential impact for treatment on a person's mobility at home in real time. And we're also using new techniques such as artificial intelligence to interpret patient data, and this can inform future trial design. For example, artificial intelligence, so machine learning analysis of anonymized trial data and real-world patient data, can better identify the patient subpopulations or subgroups in our trials and to try and determine what factors are affecting their treatment response. So during the approval process, regulators like the MHRA assess data from trials and other studies of a treatment to determine its safety and efficacy, and then that's to decide whether to grant it a license. And then health technology assessment bodies such as NICE assess its cost effectiveness to decide whether it can be made available in the NHS. So they need to assess different types of patient data, for example, how common or rare a condition is, how it changes over time, how it affects your everyday life, and how the costs of care and current treatments are presenting themselves, and also the potential value of a new treatment. So to provide all this evidence, at Pfizer we're conducting studies of anonymised real-world patient and caregiver data, working together with patient organisations and healthcare professionals. And we're also collaborating with other initiatives like Project Hercules that was set up by Duchenne UK to build a better evidence base for Duchenne. But even after approval, ongoing patient data collection can be needed, and that's needed to understand a treatment's longer-term risks, benefits, and cost-effectiveness. So at Pfizer, we work closely with multiple stakeholders, including national registries, to enable this. So in conclusion, patient data are essential to Pfizer to understand and demonstrate how conditions and treatments can affect people's lives and to bring more effective treatments more quickly to patients and their families.
Thank you very much. So we will move on to the next question to Roche. The drug development doesn't really stop with the phase 3 trial. Ultimately, we want to get access to better treatments. Could you explain the progress that it takes from phase 3 clinical trial to actually market approval, and a bit about the timelines as well?
Yes, thank you very much, Professor Straub. I'm Roberta, and I'm a medical affairs partner with Roche UK. So we heard a lot today about clinical trials and data. So once the clinical trials are over and the company has the data, the first thing we do is take some time, and this could take a few months, to analyze the data. That analysis of the data is then included in the dossier which we submit to the MHRA and also to NICE. And Emily yesterday explained very eloquently the roles of MHRA and NICE, so I won't be doing that myself today. So MHRA will be looking at the dossier that we submit to them, and they will be assessing the clinical effectiveness of the drug. They look at the safety profile of the drug. They also look at the quality of the drug. And what they are really intent on achieving is that the benefit-risk profile of the drug is a favorable one, i.e. that the benefits of the drug outweigh the risks for the patient, so the side effects, the potential safety profile that the drug could have. NICE look at it from a bit of a different perspective. They also look at the clinical trial data. However, they look very much at the cost effectiveness of the drug. So economic modeling is a really important part of the dossier that we submit to NICE. And these could happen in parallel, although the NICE approval is always subject to the MHRA license. So you won't get a NICE approval if the drug isn't first licensed by MHRA. But because both processes are lengthy and take months, and I can't give you definitive timelines, unfortunately, because it all depends on the complexity, very often what we do is we try to do them as much as we can in parallel. Now, what NICE do as part of their process, which I think is probably the part that will interest you most over here, is they engage very much with the external stakeholder group. They engage with clinical experts, they engage with patients. And the reason that they do this is because, as we heard yesterday as well, patient evidence and input are an important part of the process, especially in rare or complex conditions such as DMD, because you can provide unique insights into the impact of a disease and the treatment on everyday life. As I said, it's very difficult to assess the timeline that this would take, but roughly speaking, it usually takes anywhere between 18 months to up to 3 years, and a lot would depend on the complexity of the medicine, the quality of the data provided, and other factors. Thank you.
Thank you very much. We'll move on to question number 7 and to Santhera. And so the question is, what do you need to demonstrate to regulators, and we have just heard about HTA agencies, to achieve a drug's approval, and what types of approval could a new drug receive?
Thank you. So good afternoon, everybody. Thank you for having us. I'm Elodie Danjean. I'm the General Manager in the UK for Santhera Pharmaceuticals, as of yesterday. Coming back to the company, I'm delighted to be back to the field. So the first thing, and I apologize because there's a little bit of overlap with the previous question, but basically the first thing that companies need to demonstrate is clinical effectiveness, that this product shows a benefit either against placebo or against the standard of care. So the concept of standard of care is really important, especially in Duchenne. There are already standards of care that are now being captured in DMD care, etc. And so what we as companies need to demonstrate is not only that the product works, but actually that it presents a benefit over and above the standard of care. As well as demonstrating, of course, that it's safe enough. No product is entirely safe, but it needs to be safe enough, and that we can manufacture it to acceptable required quality standards. So it's quite a complex, structured approval, but that's the core of the product. And I think it's important to emphasize the piece around it needs to be effective enough, safe enough, and adhere to the quality standards. No product is perfect, as unfortunately you know. So that's the first stage. And the second piece that we need to demonstrate is not only is it clinically effective, but also it is cost effective. So it makes sense for society and taxpayers' money to be used because there is a clear benefit, and that spend on this product provides a clear benefit to patients, to their carers, and to society at large. And so that's the second stage, which is normally undertaken by HTA bodies. In the UK, we have NICE and SMC. And so very, very different questions will be asked about cost effectiveness, and the conversation between the company and the payer will be around, in NICE in this case, which is not a payer, but a HTA body, will be around based on a cost effectiveness model. We're very fortunate in Duchenne that Project Hercules has delivered a cost effectiveness model that companies like Santhera can use. And so the conversation will be around, we'll be using very technical concepts such as ICEs, which really try to quantify the delta of cost, the ratio of incremental cost versus incremental benefit of a drug. And they use this as criteria to evaluate the cost effectiveness of the product. So it's not a case of a monologue, like a one-way street where we say that's the model and NICE says yes or no. There's a lot of conversations between NICE, the companies, and patients are involved, and so are clinicians in these conversations.
Thank you very much, Elodie. Oh, can you hear me? Next question to Sarepta, please. Could you comment on the reasons for choosing a specific patient population to take part in a clinical trial, and how the data is then used to inform which patient population may benefit for market approval and access purposes?
Of course. So hello, everybody. My name is Jared. I'm coming from the medical team at Sarepta. And I'd just like to say congratulations to Emily and Alex on what has been an amazing event, and also really well organised and forward-thinking. So we cannot wait to be back again next year. So for our initial exon skipping trials, we wanted to include a broadest population as possible that were amenable to exon skipping. And then we chose a part of the population that had really good natural history data available and endpoints and assessments that were highly regarded by clinicians and patient advocacy groups that we work with. And then after that, we want to really examine that and make sure in our clinical studies we can incorporate those. That's what we ended up choosing. And then I wanted to say that we didn't have any intention to develop therapies just for young individuals with Duchenne, but all individuals with Duchenne. And you'll see now we have trials looking at older individuals, and we're assessing, for example, the heart, the lung, and also looking at the performance of the upper limbs. And this is really an exciting time in the Duchenne community. Also, we take learnings from every single trial that we run. So, for example, every trial we can get feedback and incorporate that into every single protocol. And as you know, we engage community advisory boards. We have Caroline Daly and our patient advocacy team. We get meaningful feedback and we incorporate those in every single study that we do. So for market access and approvals, I mean, it's a really hot topic at the moment. I think regulators, they're listening to companies, they're listening to patient advocacy groups, they're listening to experts in the field, and they are becoming more progressive. But as you all know, in a disease such as Duchenne, which is progressive, and time is muscle, they could do more to be more progressive. And for example, we'd like to see regulators, for example, the MHRA here in the UK or EMA in Europe, potentially accept surrogate biomarker data, or also to exercise the highest degree of regulatory flexibility in a disease like Duchenne, where there's a really high unmet medical need. And I'd just like to say thank you very much for listening, but thank you for all of you who've been involved in our clinical trials throughout the process, not just our trials, other companies' trials, any studies, and this is really pulling the community forward and helping us better understand this condition. Thank you.
Thank you very much. Our next question is for Solid Biosciences. What have you learned from your previous development programs in DMD, and what are your plans moving forward?
Thank you. Thank you for having us here. And just to echo, thank you for all of the families and the patients that have contributed so much. So my name's Gabe Brooks. I'm the CMO of Solid Biosciences. And Solid Biosciences has a long and enduring dedication to developing clinical therapies that make a significant clinical impact on patients with Duchenne muscular dystrophy. And as the question alludes, we did have a prior therapy in clinical trials. After looking at 9 patients dosed with that study, we actually discontinued those therapies. We did see some signals of effect, but neither the microdystrophin expression nor the clinical efficacy were significant enough to enable us to continue that study in the setting of actually seeing some safety events in terms of inflammation. Now, this program was one of the first programs to dose high-dose AAV gene therapy, and the inflammation and safety signals we saw have been recapitulated in other clinical trials. So this is really a learning from the field. And at Solid Biosciences, we've looked very carefully at what we thought was underlying those safety signals, and we're really proud and pleased that we were able to collaborate with our industrial colleagues as well as academics. And really, specifically, we're quite thankful for being one of the founding members of the consortium that's run by Dr. Francesco Montoni and Karsten Bonnerman. That has been looking at safety in AAV gene therapy. So now we are coming back into the clinic with a next generation gene therapy for Duchenne muscular dystrophy, and we went back to the drawing board and looked at all the components to say what would make an improved approach to these patients. And so we maintained our initial microdystrophin, which includes a critical aspect, which is nNOS localization to the sarcolemma, because we believe that can improve muscle integrity because of the compensatory vasodilation that you need with healthy muscle. We then, we understood some of the safety signals were due to dose of capsid, of antigen. So we wanted to find a novel capsid that can reduce the dose required, and in preclinical models, we found a capsid that robustly induces muscle, and we hope, and this is why we're engaged in the work, to see if this can improve expression in patients. And then finally, we took learnings about the production of AAV, and now we have a new process where we hope that we can reduce the amount of excess capsid for improved efficacy. Thank you so much.
Thank you very much. We'll come to W. So Francesco, it does work, I mean, to Wave Life Science. And so the topic is slightly different here, the question. It's about cardiomyopathy, so in the heart, and skeletal muscle as well. The disease starts at birth. And cardiomyopathy is a major cause of early death can you comment on the challenges of developing treatments for the heart in Duchenne from an industry perspective?
Sure. Thank you so much for the question. And hi, everyone. My name is Shelley Casey. I'm the head of advocacy at Wave Life Sciences and appreciate the opportunity for being here with you all today. So I think it's a very important topic and not one that doesn't come with a few challenges. So what I wanted to do is just share a few of those challenges with you today. One of the first ones is really thinking about the patient population that's included in clinical trials. As we heard yesterday, the heart is affected from the start and symptoms start late. And typically in clinical trials, it represents or includes a younger patient population. And some of the cardiac measures really might not apply to that population being studied. So that was number one. Number two is the lack of global cardiac treatment guidelines. So treatment is applied differently, which could potentially confound results or lead to a longer period of time to really help standardize treatment prior to patients starting trials. And third, availability of cardiac MRI. So one of the measures that we look at is ejection fraction, which is generally measured by either echo or cardiac MRI. And in general, there's about a 1 to 3% loss of ejection fraction year over year. And so echo is typically more common, but the challenge with echo is that there is a variability of about 5 to 10%. So if you're trying to tease out just a difference of a few percentage points, it might be difficult to see that change. Cardiac MRI, on the other hand, is much more accurate within about 1%. So it is the gold standard. I think people look at it for its precision and its reproducibility, but the challenge is it's not available at all sites and all centers. So in summary, I think there are a few challenges, but as we heard earlier today, the goal is to create data that's robust and reproducible. And there's a lot of ongoing work streams that are currently underway to really try to identify ways to be able to measure the impact of these investigational therapies on the heart. So we're very supportive of that. Thank you.
Just to hold it. Thank you. No, thank you very much for all your answers. Also, thank you very much for excellent timekeeping, sticking to your time. I apologize to Abby that you were the only one who I cut short, despite the fact that we still have 1 minute left. So, it's fantastic to see, as I said, the number of companies. I think it's also amazing to see the expertise that these companies bring into the field, and that also big pharma, like the global players are involved in what is still a very rare disease and a niche. So thank you very much for willing to be on stage here and for answering these questions. Great.